Spotlight on Antibodies, a podcast from Biointron, where we bring you the latest antibody industry trends. I'm your host, Jovina Celestio, and this month in March, top news includes ion channel and GPCR-driven diseases, affinity maturation, and a look back at SARS-CoV-2 5 years on. G-protein coupled receptors and ion channels are protein families linked to a broad spectrum of diseases, known as GPCR and ion channel-driven diseases, which arise from dysfunction in cell signaling and ion transport. GPCR-driven diseases include conditions such as cancer, metabolic disorders, and neurological diseases, like migraines, while ion channel dysfunction is implicated in epilepsy, chronic pain, and cardiovascular diseases. While small molecules have traditionally dominated this space, advancements in antibody discovery have paved the way for more selective and durable treatments. With breakthroughs in antigen preparation, structural biology, and screening technologies, monoclonal antibodies against GPCRs and ion channels are progressing through clinical pipelines, promising new solutions for these challenging disease areas. A few weeks ago, Maxion Therapeutics, a UK-based biotechnology company, raised $16 million in pre-Series A financing, with the aim of developing antibody drugs targeting ion channels and GPCRs using their NotBody platform. Their platform fuses knot-ins, which are naturally occurring cysteine-rich mini-proteins, onto antibodies to enhance drug properties. Ion channels and GPCRs are critical cell surface proteins involved in a wide range of previously untreatable or poorly treated diseases, including autoimmune conditions and chronic pain. While small molecule drugs exist for ion channels, no approved antibody drugs currently target them. Meanwhile, a recent review describes the discovery of therapeutic antibodies targeting GCPRs and ion channels as complex multi-spanning membrane proteins which are embedded in the cell surface lipid bilayer by multiple transmembrane spanning polypeptides. Antibodies that bind to the selected targets may directly modulate protein function or be reconfigured into formats such as ADCs or bispecifics, which enable alternative modes of therapeutic intervention. However, these targets present challenges due to difficulties in high-level expression, extraction, and stable antigen generation, requiring diverse discovery strategies, including hybridoma, B-cell platforms, and display technologies. Emerging approaches involve novel antigen formats, alternative hosts, and microfluidic platforms. Despite progress, no clear consensus exists on discovery methods, making antigen generation a bottleneck. Advances in structural biology, computational modeling, and machine learning are accelerating antibody discovery with the potential for fully in silico designed antibodies, paving the way for new therapeutic approvals. Allosteric antibodies are also emerging as powerful tools for targeting traditionally undruggable membrane proteins, including GPCRs and ligand-gated ion channels, by stabilizing specific conformations to modulate activity. Despite GPCRs being major drug targets, antibody-based therapies remain scarce due to their small extracellular domains and complex structures. Recent breakthroughs, including VHH-based modulators for mGlu receptors and calcium-sensing receptors, demonstrate their potential in neurological and metabolic disorders. Similarly, allosteric antibodies targeting LGICs like alpha-7 NHCHRR and NMDA receptors have shown promise in enhancing or inhibiting receptor function. Advances in AI-driven design and cryo-EM are accelerating allosteric antibody discovery, offering new avenues for therapeutic and drug discovery applications. Affinity maturation is a fundamental process in adaptive immunity, enabling B cells to refine antibody specificity and enhance binding strength through iterative cycles of somatic hypermutation and selection. This natural optimization mechanism is not only necessary for effective immune responses, but also serves as the foundation for antibody engineering in therapeutic development. Recent research has expanded our understanding of affinity maturation, revealing intricate biological regulations, novel computational strategies, and innovative methodologies to enhance antibody binding. The germinal center is the central site of affinity maturation, where B cells undergo rapid division and SHM to improve antigen binding. However, because mutagenesis is random, excessive mutations can be detrimental, potentially disrupting beneficial antibody structures. A recent study investigated this phenomenon, proposing that high-affinity B cells mitigate this risk by reducing their mutation rate per division. Data from mice immunized with SARS-CoV-2 vaccines and model antigens confirmed this hypothesis, showing that B cells producing high-affinity antibodies shorten the G0, G1 phases of the cell cycle, allowing for more cell divisions but fewer mutations per cycle. This finding highlights a key regulatory mechanism that safeguards high-affinity B cell lineages and optimizes the efficiency of affinity maturation. While affinity maturation is typically driven by antigen binding and T follicular helper cell selection, external factors can influence this process. A study led by UCL researchers examined the effects of monoclonal antibody infusion on affinity maturation and found that high-affinity monoclonal antibodies can suppress SHM and reduce the affinity of most competing B cells. However, a subset of B cells exhibited increased mutation rates and enhanced affinity, suggesting that exogenous antibody infusion can apply selective pressure that accelerates the maturation of specific epitope-targeting B cells. This study provides valuable insights into how therapeutic antibodies or vaccine strategies could be designed to shape immune responses for improved protection. Beyond biological mechanisms, computational models are transforming antibody optimization. A recent study published in Nature introduces GPCR-driven, a geometric graph neural network designed to enhance antibody affinity through deep learning. By leveraging multi-relational graph construction, geometric message passing, and contrastive pre-training on large-scale protein structural data, GPCR-driven significantly outperformed traditional approaches. This model successfully improved antibody affinity, with ELISA EC50 values decreasing by up to 17-fold and KD values by up to 6.1-fold in designed antibody mutants. These findings underscore the potential of AI-driven methods in predicting and enhancing antibody-antigen interactions, reducing the need for labor-intensive experimental affinity maturation. Another breakthrough in computational affinity maturation comes from affinity flow, a machine learning framework that optimizes antibody affinity using only sequence data. Unlike structure-based models, affinity flow employs flow matching to generate diverse protein structures from sequencing, followed by an alternating optimization framework that iteratively mutates antibody sequencing for improved binding. A key challenge in developing such models is the scarcity of labeled training data. To address this, Affinity Flow incorporates a co-teaching module where a sequence-based affinity predictor refines a structure-based predictor, and vice versa. This approach has achieved state-of-the-art performance, demonstrating the power of AI in streamlining affinity maturation without relying on costly structural experiments. Affinity maturation remains a cornerstone of immunology and therapeutic antibody development. The latest research highlights how B cells naturally regulate mutation rates to maximize affinity gains while minimizing detrimental changes. At the same time, selective pressures such as monoclonal antibody infusion can shape the trajectory of affinity maturation, presenting opportunities for vaccine enhancement. Meanwhile, the integration of artificial intelligence into antibody engineering is revolutionizing how we approach affinity optimization. As our understanding deepens and computational tools become more sophisticated, the future of affinity maturation promises not only greater insights into immune system dynamics, but also advancements into antibody therapeutics. 6 years after the emergence of SARS-CoV-2, the landscape of viral neutralization has shifted significantly. Advancements in antibody therapeutics, vaccine development, and immune response understanding have reshaped our approach to combating COVID-19 and its evolving variants. As researchers continue to refine strategies for long-term protection and pandemic preparedness, key questions remain. How effective are current neutralizing antibodies against new strains? 2 weeks ago, a study led by researchers from Stanford University introduced bispecific antibodies that target both the RBD and a conserved region in the amino terminal domain of the SARS-CoV-2 spike protein. By integrating these two specificities into a single antibody, they developed a promising therapeutic candidate capable of neutralizing a broad range of variants in vitro. One of these bispecific antibodies demonstrated protective effects in a mouse model when administered prophylactically, reducing viral loads post-infection. This study underscores the potential of NTD-RBD bispecific antibodies as resilient next-generation therapeutics against evolving SARS-CoV-2 variants. Another recent article describes a broadly neutralizing antibody against the Omicron subvariants BA1, BA2, 12.1, BA4, and BA5. The global spread of SARS-CoV-2 and its variant strains, including Alpha, Beta, Gamma, Delta, and now Omicron, pose a significant challenge. With the constant evolution of the virus, Omicron and its subtypes have developed the capacity to evade neutralization induced by previous vaccination or infection. This evasion highlights the urgency in discovering new monoclonal antibodies with neutralizing activity, especially broadly neutralizing antibodies to combat this virus. In the recent study, a fully human neutralizing MAB, CR9, bounds to an epitope formed by RBD residues. With its strong potency and specificity, CR9 presents a promising therapeutic option for Omicron infections, emphasizing the ongoing necessity of MAB discovery to combat COVID-19. Meanwhile, in the bioinformatics space, researchers published a paper in Nature about the discovery of deep mutational learning for the selection of therapeutic antibodies resistant to the evolution of Omicron variants of SARS-CoV-2. By constructing a diverse library of full-length Omicron BA1 receptor binding domains and screening for ACE2 and antibody binding, they trained ensemble deep learning models to predict antibody binding and viral escape. This approach enabled the identification of optimal antibody combinations with complementary resistance to viral mutations. Using deep learning to analyze a vast sequence landscape, researchers demonstrated a method for selecting therapeutic antibodies that remain effective against current and future SARS-CoV-2 variants, offering a powerful strategy for long-term COVID-19 treatment. Further along in drug development is Inhalon Biopharma's Phase 1 study evaluating the safety, tolerability, and pharmacokinetics of IN006, a nebulized reformulation of regdanvimab, as an alternative to systemic antibody delivery for COVID-19. IN006 is an inhaled antibody treatment for COVID-19. Regdanvimab, an IV-dosed human IgG1 monoclonal antibody directed against the SARS-CoV-2 spike protein receptor binding domain, was approved in the European Union for adults with COVID-19 who did not require supplemental oxygen and who were at increased risk of progression to severe COVID-19. From results published last month, the trial assessed single and multiple inhaled doses, with no serious adverse events reported. IN006 achieved high nasal fluid concentrations, significantly exceeding the typical IC50 range for antiviral monoclonal antibodies, while serum levels remained low. These findings support further development of inhaled antibody therapies as a targeted and convenient approach for respiratory infections. And that's all for this month's Antibody Industry Trends. To learn more insights into antibody drug news, make sure to follow Biointron on LinkedIn or at our website at www.biointron.com. Keep up to date with further episodes from Spotlight on Antibodies by following this podcast.